Everyone. Uh, on behalf of whole team, I welcome all for today's webinar in the Data Science webinar series organized by Manav Human Atlas Initiative. Myself, Dr. Shailesh Kumar, moderator of today's talk. As you all know, that the total allotted time for today's webinar is 1 hour, out of which the talk will be of nearly 40 to 45 minutes. Then we'll have a question answer session of about 15 minutes. I request all participants to use the chat box at the bottom of your screen to post your queries. I repeat again, to use the only chat box to put your queries related to the talk. You can put your queries in between the talk, which will be answered by the speaker after the talk during question answer session. So now I would like to introduce Dr. Leelavati Narlikar. She is the speaker of today's webinar. Dr. Leelavati is currently working as a scientist at National Chemical Laboratory, Pune. She had completed her bachelor's in computer engineering from University of Pune. Then she got PhD in computer science from Duke University. As you all know, that NCL has a bunch of scientists with diverse background, from chemical engineering to applied mathematics, working on different problems in mathematical and computational biology. Dr. Leelavati is one of them. She is interested in developing probabilistic models and statistical algorithms in the area of machine learning in general and in computational biology in particular. She has largely worked on understanding aspects of transcriptional regulation, but many of her group's methods are fairly general and can be adopted by the problems in other domains as well. So today she is going to give an overview how probability theory can be used to build machine learning algorithms. So without taking too much of the time, over to Dr. Leelavati for her talk on designing algorithm to learn from the data. Dr. Leelavati.
Thank you. Thank you for the invitation and also the introduction. So today we will be talking about how high school mathematics can be used to design machine learning algorithms. So let me begin by telling you what I think of when I think of machine learning. And if you've been following this wonderful series by Manav, you probably already have a very good idea. So just bear with me in any case. So these are essentially algorithms that help the machine or the computer to learn from data to do some task, right? So for example, right, a very simple example, you might want the computer to look at an image of a fruit and report back as to whether it's a mango or an orange, right? Very simple. Of course, looking at the image of one of these fruits. So essentially, the computer has to apply some rules or some formula to these images and then come up with its prediction. So this by itself is not machine learning. So machine learning comes into play when you are when the computer itself is learning the formula, right? So you're not giving it pre-decided rules, but the computer is figuring out what are the best rules or what is the formula that it should apply if it wants to get a good score on this task, right? So naturally, if it wants to do so, it needs to have seen lots of images beforehand from perhaps different types of mangoes to make sure that its predictions are general enough when, instead of Hapu's Spirey is given, right? Or, maybe in different angles and so on, right? So it needs enough data to figure out what the rules are, right? And so essentially, these are algorithms that are going to help. So the algorithms are decided by the human, right? But the computer is going to apply them to learn from the data. And whenever you are thinking of a new problem that you want to, some new task that the computer needs to do, needs to perform, there are essentially 4 broad steps that you need to look at. So the very first one, and very important, is to understand the problem, right? And by understand, really understand the domain where the problem is coming from. And once you do that, you will be at a position to design a suitable model. And this is all on paper, right? Or in your head. And then you code it on the computer so that the computer can then learn the best parameters of the model. And this is essentially optimization. Okay? And then once you have that, your method is complete, except that now you need to test it and evaluate it and to make sure that it works. Right? And these are the 4 steps that you would typically take. This looks a little complicated, but we have been doing this as humans constantly, right? Right from when we are babies, we are essentially doing learning, okay? From what we see around us, what we sense around us, we are constantly learning different, in order to do different tasks. In fact, in many places, we are much better than the computer still, right? Which is why often when you are trying to log into Facebook or Twitter or whatever, you are, you come face to face with such a challenge, which is kind of annoying. But if it stops at this, it's good, right? You can just put in your what you think the letters mean, and just to make sure that you know what this is just to make sure that the server is trying to make sure that you are a human and not a bot, right? You're not trying to exploit the server. So that's the idea behind giving this CAPTCHA exercises to the user. So the assumption is that if you have a machine doing this, you're not going to get it right? Except that now, algorithms are getting better and better at learning handwriting and also these kind of distorted letters. So now we have kind of switched to some really annoying kind of tasks that we have to do. Right? You have to select images that contain, in this case, it's a cat, but it contain traffic signals and cars and whatnot. Right? And so the idea is that you have to look at these images and sort of pick the ones that you think match the cat. Right? And you just go on and do this, and you think this is a harmless little thing. I mean, it's still harmless, but you just think that you are, this is something that the server needs to do. Well, it's true, but oftentimes the server is also using you to annotate some of its own images that it does not really have the labels for. So for example, this top left corner animal, it may not know beforehand whether it's a cat or not, but based on your other sort of labels, you might get the other cats correct. And then because you're labeling the top left also a cat, it now knows and it can now update its own database. So you're sort of helping it update its database. This also happened with CAPTCHA before, right? When we used to get 2 words. So anyway, I kind of digress. But the point is that we are very good, okay, at these the steps that we want the computer to do. And the reason we are good is that because we have seen lots of cats before, right? And we know what they look like. What happens when you're given images like this or tasks like this? So if you're a radiologist, you probably can answer this question. I'm not, so I can't, just looking at the image, right? A computer now can be trained fairly well to answer such questions. Not just TB, right? I mean, cancer, and not just X-rays, other kinds of radiology images. Still, a radiologist does, a trained radiologist does very well, probably better than a computer. I don't know the latest, what is the latest in the field. But the reason they do that is because they've seen a lot of images and they've been trained this way. And it's the same thing for the computer. If it's seen enough images, it knows because of the algorithm that we are feeding it. So far, that's good. We're just trying to do what we can do using a computer. What about this? Right? So the question here is this piece of DNA bacterial or human? So that's a little hard. It's even harder if you're an undergraduate, the way I was, and I had no clue what DNA was when I was an undergrad. So let me just tell you what that is. Okay, this goes to understanding the problem. Okay, so DNA is essentially a long chain of nucleotides, or it's a polymer essentially, made up of repeating units of A, C, G, and T. So for a computer scientist, it's just a long string, a sequence of A, C, G, T. In nature, it appears as a double-stranded molecule or a double helix. And essentially, these are two strands which are complementary to each other in a very specific way. So an A is always going to pair up with a T, and a C is always going to pair up with a G, and that's how you get your complementary strands. So essentially, knowing the sequence on one strand gives you enough information about what's going to happen in the second strand, right? That doesn't add anything. So to a computer scientist, I repeat, it's just a long string of ACGT. Okay, all right. So that's a little bit of background about what DNA is, but still, doesn't put us in a position to answer this question just looking at the data. Okay? So that might be because we haven't seen enough. Right? We've probably never seen bacterial DNA. I mean, I have not seen much of it. What if I do give you a lot of bacterial DNA and human DNA? What then? So we'll see whether you can look at such data sets and develop methods using probabilities to distinguish between bacterial and human DNA. Okay, so probability. So this is something that we use often, right? When we are day-to-day life, we use the word probability because it's something very natural to us. It's a number essentially between 0 and 1, or between 0 and 100%, depending on how you're normalizing. And it essentially reflects your belief in some event happening, right? So what's the probability that it's going to rain tomorrow, and so on? Very simply put, it's a very simple question, is let's say you toss a coin. What is the probability that you will get a head? It's about half, right? And which is the reason why you use a coin toss to decide who bats first in a cricket match, or who's going to serve first, and so on. Right? So that's about, I think everybody's on the same page when I say it's about half, right? So if I toss a coin once, the probability of getting a head is half, right? So what happens if I toss the same coin 5 times and I get a string, I get a set of outcomes, right? A string of outcomes, sequence of outcomes, sorry. A head, tail, head, head, head. What then is the probability of seeing this data? Okay, so this is now our data D. What is the probability of seeing this? Well, the first time that you get a head, its probability is half. And then you toss the coin again. It's multiplied by half because these are independent events. And if you remember from high school, if you have independent events, then the probability of the joint or both events happening is just the probability of multiplication of the two probabilities, right? Because the occurrence of the tail is independent of whether the first throw was a head or not. And then so on, right? And so you multiply these 5 terms and you get 1/2 raised to 5, right? That is the probability of seeing this data when you have this coin. What if it's a Shailesh coin? So I guess I'm talking to a very young crowd here. So there's a chance that you've never seen Shailesh. Hopefully you know the 2 characters on your screen. If you've not seen the movie, make good use of the lockdown, watch it. And if you're going to watch it and you've not seen it before, I apologize because I'm going to talk about a little bit of a spoiler. There's a coin in here which always gives the probability of 1. Okay, essentially it always gives a head. So I might be, my knowledge about, or my memory about Shailesh might be a little rusty, so it might be tail, right? But let's just say, for reason's sake, it always gives a head, right? So on both sides you have a head. In that case, the probability of seeing the previous sequence of events is 0, right? Because there is no possibility of a tail in that case. But what if it's a slightly biased coin, yeah? Which means it's not half, but it's, let's say, 0.8. What happens then? First of all, how do we represent this now bias? Okay, So we can represent this model, so to speak, as a Bernoulli probability or Bernoulli trial. Okay, so we're saying that the probability of coin giving a head is P, which means that the probability that a tail comes up is 1 minus P, right? And in a fair coin, it's half and half, right? It's equal. In a Shailesh coin, it's 1 for head and 0 for tail. And if you have a slightly biased coin, your graph, your probabilities might look like this. Okay, so now you have a model, okay, which is a coin with a probability P when you toss it of getting a head. All right. So when you had a fair coin, this was your probability from the earlier slide. What happens when your coin is now biased with this probability? So not, the formula remains the same. The probability of a head is now 0.8. Now the tail probability is 0.2, but they're still independent events because it's still the same coin that you're using with the same probabilities. And this ends up being essentially formula is the same. So P raised to the number of times you see the head times 1 minus P raised to the number of times you see the tails, right? This is just a compact way of writing these probabilities. So if you have any questions, I know you're not supposed to ask in between, which is a bit of a pain in these kind of webinars. But if you just remember the slide number, we can get back to it at the end.
They can ask questions, means they can put the question there in the chat box.
Yeah, correct. But I may not see them in time. So just make sure that you put in the slide number so it's easier to get back.
Sure.
Yeah? All right. So this is the general sort of formula. And if you've seen this before, it's too easy for you. That's fine. Good. I'm happy. And this ends up in this case being higher. Okay. So now, if you had to determine whether the coin that was used to get these throws was a fair coin, or the slightly biased coin, or a Shailesh coin, well, you would kind of hedge your bets on it being a biased coin, right? Because it's 0.08, it's higher. Okay, so what you are now doing is essentially predicting. If I didn't tell you which coin I used, but I told you that the coin was one of these three, you could then start guessing, predicting which coin it might have been, right? So how can we use the same approach for this DNA? So now, if you remember our bacteria, imagine the bacteria is coming from one coin, and DNA is coming, human DNA is coming from another coin. Except that it's not head or tail, right? Because you have 4 possibilities now. You have A, C, G, and D. So you have to look at the slightly more general generalization of the Bernoulli, which is a categorical distribution. So instead of 2 possibilities, you have now K, in this case, 4 possibilities. And the 4 probabilities now have to sum to 1. So it's like a die, right? A die has, when you play dice, sure you know what a die is. It has 6 sides, and you might get 1 of 6 when you throw, right? In this case, you're getting 1 of 4 possibilities. And imagine that, you start sort of playing God, and you're tossing a coin with 4 sides now in ACGT, and you're using the same coin to decide which the next nucleotide should be. And you can sort of model your bacterial DNA in that format. And if I tell you beforehand that, just like I told you about the coin, which is slightly biased, I can tell you that bacterial DNA has, let's say, this bias, and human DNA has the second bias, which is, more GC rich, which is not true, by the way. Okay, these are fictitious values that I've just cooked in my head. Okay, so this is not real, but it's just an example. So then you can score your sequence just like we did for the earlier coin case and decide whether it comes from a bacteria or a human. Okay? But this is still not machine learning. Okay, you might be good at predicting, but it's not machine learning, because this comes back to our hard-coded rules, because I'm telling you beforehand what the coins are, right? I'm telling you the biases. So essentially, you need to learn these P values, right? In this case, you need to learn the categorical distribution values of the DNA. How do you do that? So let's go back to the coin, because it's slightly easier to put in. So naturally, to learn anything, you need a lot of data. So in this case, let's say you ask the person whose coin you're using to toss it lots of times. And then you just count the number of heads, right? So let's say K is the total number of heads you see. N is the total number of observations, right? That's the size of your data. And P is the bias of the coin, which you don't know, okay, which you want to find out, you want to predict it, which would now become your rules, which you can use for further predictions, right? But you do know that if you use the variable P, you can write the probability of seeing this data. Now, this is conditional, right? The line in between, which I used earlier also. It just means that given that you know your P, it's just P raised to K times 1 minus P raised to N minus K, which just follows from the number of heads and number of tails. And what you could do then is maximize this value, okay? And overall possibilities of P and report that P. And that's one way of figuring out what your bias of the coin might be. So if you decide to plot this expression, right, over all values of P, you get something, and let's say N is 100 and K is 70, right? So you tossed it 100 times and you saw 70 heads. This is what it looks like, okay? On y-axis is this probability P raised to K to 1 minus P raised to N minus K, and x-axis is P, which can only range from 0 to 1. And you'll see at 0 and at 1, the value has to be 0. And if you look at the max, it ends up being at 0.7. And that is something you might expect, right? Without knowing any of this math, that if I tossed it 100 times and I got 70 heads, you might kind of guess that the bias is 0.7. So the next time I toss it, you would expect a head to show up with 0.7 probability. And what you're really doing here by maximizing it is you're maximizing this function, which is also called the likelihood. Okay, what is the likelihood of seeing this data? And when you find the maximum of this, it's called a maximum likelihood estimate, okay, of your unknown value. In this case, it's only one unknown value, P. And you don't have to plot it each time. In this case, you can use your high school calculus to find the derivative, right? You want to find the maximum of a function, you find the derivative, you equate it to 0. It's either a minima or a maxima, if you remember. And then you can figure out what the maximum is. It ends up always being K by N. Okay? And here you can see pictorial. So the idea is, and this is what is used in many machine learning approaches that use probability theory, is do some sort of optimization to figure out, to optimize this, some sort of a function that looks like this. Okay? It's not always likelihood, there might be variants of that. Oftentimes you cannot do a differentiation and equate it to 0 and get it. Sometimes you have to write a very different special routines, which is where optimization techniques really come into play. But I'm not going to focus on that in this talk. But if you're interested, do write back. There's a lot of interesting stuff in those areas. All right. Okay, so from back to bacteria and human, you can then, once you have a lot of data, learn these values. Right? Again, this would be very similar to K by N, the number of times you see the A appear divided by the total and so on. So the formula remains very similar to the coin formula. And then you can use it to predict. So you can actually just do this on your own. Yeah? So if you like to code, you can download bacterial DNA genome from NCBI or wherever, and the human, and you train it on maybe one chromosome of human and bacteria, and then predict it on some other some left out set and see how it works. Chances are, if you know a little bit of biology, it's not really going to work very well, because what you're assuming here is that it's the same coin for each position of the bacteria or human, and that's not really, doesn't really work because our DNA is not homogeneous in that sense. Yeah, there will be regions which are more GC rich, regions which are more AT rich, and then there are also, correlations between nucleotides that appear one after the other. So these approaches may not always work, but it's a start. Okay, that is will give you, a minimum what you can do. Definitely better than eyesight. Okay? Let's see how you can combine now different probabilities. Here we've just used one coin. Okay, but you can really do really powerful things if you assume you have multiple coins. Okay, so I'm sure you have dealt with emails like this, right? Or something like this, which are annoying spam emails that if you have a good spam filter, might never let you get it, but you might end up seeing such emails anyway. And so what is the spam filter really doing? It's figuring out that there are some words in your email, right? So it's saying, dear sir. To me, sir is a red flag, because anybody who knows me would not address me like that, right? And then there's some URL in your email, and then some iPhone claim one, and these words are probably red flags. On the other hand, legitimate mail also might have some of these emails, right? For instance, I said sir is not likely, but I do get legitimate emails addressing me as sir, because, in my profession, that's an educated guess to address me as sir if they don't know me, right? I get that often. And then, you might still see some words that appear. So it's not each word itself, but it's really a combination of the words that are telling you that this is a red flag, right? The first one is spam, right? So many spam filters actually rely on something that we used before, which are coins-like structure. So you have a coin for every word, yeah? So this is also something you can build on your own if you like coding. In fact, you can just look at all emails. You can create your fictitious emails and so on if you like as well. But if you look at your spam folder, you have a lot of data in there. Okay. So what you do is you extract features now, okay, into something that is, so you create a table for every word. You just note down whether the word is present in your first email, not present, and so on, right? So yes, no, yes, no. It's like a head or tail. And you do this for all words except maybe the common words, right? The, o, and so on. And then for every word, you can compute the probability that that word is present, given that it's a spam email, right? When you know that it's a spam thing, right? When you know that it's a bias, that it's a special type of set of emails, and you do that for every word. So each word independently, right? So that's an assumption that you're sort of not looking at what other words are in there. Each column you're doing on their own. But you are essentially using D different coins. You use the same coins, same words, and you create a table for your legitimate emails as well. Okay, and you get a similar set, but you get a different set, hopefully a different set of probabilities. So you might get something like this after doing all that. When you compute the probabilities, how would you compute the probabilities? You know how to do maximum likelihood estimate, right? You just have to do K by N. You do that. You'll notice that some words are definitely different distribution in spam versus legitimate. As on the other hand, the last word, has no, doesn't really distinguish between a spam and a real email. All right, that's good. And then what you can do is you can combine. Sorry, wait, hold on. Let's then you get a new email, right? And the new email now you parse using the same features of your words, and then just say whether the word was present or not. And then you compute the probability that it came either from the first table or the second table by multiplying them all. Okay, that's an assumption, and we can talk about that later, but you're assuming that they're independent once you know that it comes from spam or not. And then you just, based on whichever value is higher, you assign it to that class. And that's how many spam filters really used to work for a long time. I think they still do. And it's something that you can try out on your own if you like coding, instead of using a package. All right. So let's go back to our 4 steps. Okay, so we said we have to understand the problems, which we did for the spam filter. We also want to therefore using that design a model, good, and then learn parameters and test. We didn't talk a whole lot about testing, but because we don't have a whole lot of time, but we can get to it later. So let's. I'll show you one little story in the last maybe 10 minutes that I have, where we used a similar sort of approach in a biological problem. Okay, and I always talk about learning rules or formula, but sometimes, when you're in the process of doing this, when you're using a probabilistic model, you can also gain insights into your data, which you earlier did not know. Okay. All right. So let's go back to DNA a little bit. Okay. If you remember, DNA is a double-stranded molecule, and cells store their hereditary information on these molecules, and the whole DNA content is known as the genome of that organism. We now have the whole, human genome sequenced, right? Which is essentially lots of letters. Okay, we have 23 pairs of chromosomes, and if you just look at one set, the one that you get either from your mother or father, it's about 3 billion letters. Okay, so that's a lot of data, a lot of nucleotides that you are looking at. And that's good. And we now have the sequence of these chromosomes, right? So you can go to any genome browser, for example, the UCSC genome browser, and put in a section of some chromosome and view it. Right? You can zoom out, you can do lots of things. You can see the nucleotide sequence, etc. You can also see what are known as genes, right? Because that is what is a critical part of the DNA, right? Which stores genes. So parts of the DNA code for genes, and there are lots of parts which don't code for genes, and we'll see what those are doing. But what's a gene really? So gene is just a section of the DNA, and when it needs to be used, it is first transcribed to create an RNA molecule, and then translated, if it's a protein coding gene, into a protein molecule. Right? And if you don't know what they are, that's fine. All you need to know is that it is these proteins and these RNA molecules that are actually doing all the work in your cells. Okay, so DNA is essentially just a recipe book, okay, that has all these recipes to create your proteins and your RNA molecules. But only when the recipe is actually followed out, the cell is really going to be able to use the protein or the RNA. And if you think about it, all our cells have the same DNA, and therefore they have the same recipe book of all these proteins and RNAs. But we have a lot of cell types. Okay, so if you look, these are only a few, and you can see that they look very, very different. And they also do very different things. The function is very different as well. And like I said, all the work is done by the proteins. Right? So what this means is, what is really changing, if we know that the genome is not changing across cells, what is changing is the protein and the RNA content, or the distribution of these molecules, because the recipe book is the same. Right? So what is really happening is that in a particular cell type, you might have a lot of gene A and no gene C, while in another cell type, you might absolutely not want gene A, but might want gene C. Right? So the cell is actually controlling all these steps to ensure that the final product is sort of what is required in each cell. And when things go wrong is when you get diseases and disorders. And a lot of this regulation, so to speak, happens at the transcriptional level. Okay, so deciding whether the RNA transcript is to be created or not. So let's just talk about how that happens, and then I will explain the problem statement that we were trying to solve. Okay, so let's say this is still understanding the problem. So bear with me. If you're looking at it from the computer science or the data science point of view, you have to really understand the problem. And if you're a biologist, then this is all something you already know. So hang on for the math. All right. So you have your genes. I said. Of course, you have genes in your DNA, but there's a large section of your DNA that does not contain genes. Some parts of this of this non-coding DNA harbors tiny signals, okay, that are recognized by special proteins that are called transcription factors. So these are tiny boxes, are transcription factor binding sites, where the protein, if it is present, can come and bind. Okay. Now, let's say you're looking at the heart, okay, some tissue heart. And you have a set of transcription factors or proteins that are going to be present in that tissue at that time, right? Now, because not all proteins, remember, are present in all cells, right? So in the heart, you're going to have a small subset of all possible proteins. And if the binding sites of these proteins exist on the DNA, they're going to, because they're around, they might be bound by these proteins. And if they're located at the right position, this ends up causing it might end up causing some sort of a conformational change to the DNA or the chromatin, and causing the gene to be expressed. And if this happens, we call these regions, okay, enhancers, because they are activating the genes. And this is an enhancer that seems to be specific to the heart. The reason I say it's specific is maybe this gene needs to be expressed in the brain as well. But in that case, there might be a different set of proteins and a different enhancer that might be active at that point in time. So we and a lot of biologists are interested in identifying all these enhancers and these little boxes that might be critical for expression of genes in different tissues. Why? First of all, while we are understanding the problem, we should also understand why it's important to do this. Like I said, if you don't have the correct proportions of your protein in the cell, you can end up having diseases and disorders. So you might have a perfectly good gene, but it just may not be expressed if you have a mutation in one of these boxes, right? Because the enhancer then is no longer active, okay, and it's no longer able to help express the gene. So the gene might be there, but the enhancer is not. And so the gene does not get expressed. And so by express, I mean the protein is not created. And so we and our collaborators, this is from long ago, by the way, so our collaborators were also interested in, especially in heart, okay, to understand all the enhancers in the heart. All right. So we are still understanding the problem here. Why is this a hard problem? So these regions can be anywhere. They can be very far, they can be close, they can be inside the gene, they can be anywhere. And even harder is the fact that they're not very long. These regions are tiny, okay, and they don't have a very well-established structure, unlike genes, which have introns and codons. So essentially, it was really like searching for a needle in a haystack. So, but what do we know? We know a little bit, right? First of all, before the human genome was sequenced, we had biologists and cardiologists very interested in the heart. And so they had painstakingly identified regions, enhancers, that were critical for expression in the heart. Right? And similarly, we had regions which are not. So we had a positive set and a negative set. So this is our data. And now we wanted to distinguish, very similar to our bacteria and DNA, human DNA. We had a positive set, but a positive set of human or mouse, and a negative set, again, of the same organism. So now these are regions that are enhancers for the heart versus not. Right? And like I said, a simple ACGT model does not work as well. Oops, that was not supposed to happen. Can you still see it? All right.
Yes, we can.
Yeah. Okay. We're back, right? You can see. Okay.
Yeah.
So what do we know? What can we use then if the ACGT model doesn't work? Well, we do know that transcription factors are the ones that are regulating or binding to enhancers. Right? And I said that transcription factors like to bind tiny boxes. And for some transcription factors, we already know what kind of boxes they like. So for example, here's a transcription factor that seems to like to bind all these regions. And if I color code them, you can tell that there is some pattern in them, right? And this pattern is what we call a motif that a protein likes to bind. And how do you represent a motif? Again, probabilities come into play. Okay, so we just store, again, categorical distributions. What is the probability that it likes an A in the first position, a C in the second position, and so on? And you just store it as a table. And if you're a biologist, you must have seen something like this. often enough. And this actually just follows from this probabilistic representation. The nice part about this representation is that once you have this motif, you already know the motif, you can scan and decide whether your sequence contains the motif match or not. So whether it might contain a binding site or not, right? So here's what we did. We do know, or at that time at least, we knew the motifs for many of these transcription factors. Okay, but we decided not to restrict to those that we know are active in heart, because that set is incomplete, right? Because we expected to not know everything about the heart already, right? So we said that let's say we know nothing about what is active in a heart at any point in time. Let's just take all the motifs that are present. At that time, there were about 700 well-curated motifs in database. This is like 10 years ago. And what we did was we created our feature set. Okay, for every sequence in our enhancer set and every sequence in the control or negative set, we counted, just like we did for the spam and non-spam, whether the motif matches or not. And we built a model, which I'm not going to discuss in the interest of time, but it has a nice probabilistic representation as well. Okay, and once you have that model, the model returns for any region a score, okay, a probability between 0 and 1. So you can just put the whole genome through it, and you can get these peaks. Completely computational approach, right? And it will give you, and you can then say that maybe that region in the middle is likely to drive expression in the heart. Okay. So we also we did the standard testing and validation approaches like k-fold validation and so on, which you must have heard in the earlier talks. And that kind of gave us some confidence in our approach. But the other thing we looked at was, the weights, the probabilities that we get, really the weights that we get for every feature. That means every motif. The ones that were really high were already known to be associated with the heart. Right? So this is without telling the method that we're looking for heart, without telling it anything about the heart. It picked up motifs that were heart associated, just because they were more often present in the heart set versus the not. Okay? And this was also very reassuring to us and also to our collaborators who were the experimentalists. Okay? So they were the ones who were going to do testing in the experimentally, right? Because that's where proof of the pudding is, right? How do you then check whether a new prediction is really a enhancer or not? So they made use of a reporter gene. Okay, so this is very cool. So you have a reporter gene that glows or has some mark when it is expressed, when the protein is expressed, and you place your prediction next to the reporter gene. And if that reporter, and then you put that whole thing in your organism of interest, and then see whether that gene is expressed. And if it is expressed, then you can say that your predicted region is acting as an enhancer. Okay. So we couldn't do this in humans, right, for obvious reasons. I mean, they also couldn't do it in humans for obvious reasons, but you can always do them in mice and other organisms. So you do this, you create this construct artificially, and you insert it in an egg, in the embryo or the egg, right? And then track where all is glowing. So first, our collaborators, this was done in Marcelo Nobrega's lab, were not terribly confident. So they said, let's first do it in zebrafish, which are cheaper. Okay, these are tiny fish, and they saw that the method was doing well. So you can see it's glowing in the heart. So that is, by the way, the heart, the arrow there. For those who have not seen zebrafish before, so these are zebrafish, little zebrafish, baby ones. And then once they were confident with that, they put them in mice, which are more experiments which are more expensive to do. And you can see that the heart here is glowing or has a color. And it actually did quite well. And here's a little movie where you can see the zebrafish heart is moving, is beating, and you can see it glowing as well. And it's actually, if you think about it, it's really crazy because the piece that they're inserting is a human piece. Okay, so this is a human piece that they're inserting in a zebrafish, and for some reason that piece is active in a zebrafish. So it's activated by zebrafish transcription factors, although the heart in the zebrafish is rather different from ours. It has fewer ventricles and so on. But it looks like the machinery that is active in both these organisms is kind of conserved. And it did quite well. It got some 60% accuracy in predict, so they tried out some 20 little pieces in zebrafish and mice, and about, I think, 60% of them reported back as positive, which is apparently quite good. Okay, this was done this is done when I was a postdoc in NIH long ago, right? And if you want references to this work, they're not too hard to find. So what is it that we are missing? Why is it 60%? First of all, of course, the fact that we're putting a human piece in zebrafish, there might be, of course, some problems there. But there's also this other issue that we are only looking at sequence, right? We are only looking at the sequence data, whether the binding site is there, we assume that it's going to be present and bind and so on. So we're completely forgetting that there's this other layer, chromatin, that is coming into picture. So we are just considering DNA like this as a linear molecule, but actually it's packaged tightly in the form of nucleosomes, right? So those little balls that you see are histones. They're proteins around which the DNA is wrapped. And it's really compact because it has to fit in our cells, right? So if you, I think, from what I remember, if you take all the DNA from our cells, remove all the histones, and put it one after the other, it is actually going to span the whole solar system. So it's a lot of DNA, and it is tightly packaged. So during gene expression, these regions need to be sort of opened up, because otherwise, how will the transcriptional machinery go in there and transcribe? And so there are these other factors that are sort of giving signals to these histones to move away, or trying to tell the proteins, DNA binding proteins, to, hey, come and bind here, or don't bind here, and so on. So this is an added layer, okay? This is called epigenetics, this added layer of regulation, okay? Or signaling, rather. So far, we only looked at the orange part, which is the DNA in this picture. But it turns out that this DNA can be modified, okay, chemically modified, methylated, usually. And usually in the human genome, at least at the C, okay, you remember the ACGT alphabet. And when that happens, usually the DNA is not very active at that time. Although, when I always say usually, because there are always two possibilities in biology. Similarly, the histones can also be modified, and then these modifications have effects on gene expression. So although your DNA might be the same, right, but your marks are changing, and therefore the expression is changing. And this is really critical when you think about differentiation during development, right? We all began in an embryonic stem cell, right? And but we got all these different types of cells in our body during development, and this is because of these epigenetic marks that dictated which trajectory your cells are going to take as they develop. And so the reason I'm telling you this is because there's a lot of data now coming up in these areas, because we already have the DNA, right? We have the DNA is good, but it's clear that that's not enough. We want to know what is functional and what is not functional in that DNA. So I think, okay, maybe I'll just give you one cool example from Randy Joto's lab from Duke University. So this is what they did, right? There's a agouti gene which causes a mouse to have two characteristics. They become fat, obese, and also yellow in color. So yellow is nice because you can tell that the gene is present, right? And these mother mice, if you give them regular mouse food, they give pups, which also looked similar to the mother. On the other hand, when you gave them mother methyl donor groups, okay, which are critical for dictating these epigenetic marks, the pups were healthy. Okay, so they were also not yellow. So when they looked at this, the gene, both these sets of pups had the gene, except that the gene in the healthy pups was turned off epigenetically. So they had this silencing marks on top, but the DNA was the same. Yeah, so it just was silenced out because the mother was fed supposedly a healthier diet, right? And it turns out that these same rats, when were followed for another generation, but now given regular diet, the mouse which had the gene turned off continued to have the gene turned off, although given a regular diet, no healthy diet, right? Regular mouse diet. So it turns out that, you are what your grandmother ate. So these are marks that are determined in the womb, right? But what does that mean for twins, right? Because they have the same DNA and they share the womb, so they presumably have the same marks. So it turns out that these marks can be added and removed based on environmental cues. So they've also found that mice which are under stress, right, or basically mice which are not groomed by their mothers, after they're born, end up having anxiety. Okay, I don't know how they measure anxiety in rats, but they do. And mothers who don't groom, the mothers who groom, lick their babies when they're born and, sort of love them, so to speak, those turn out to be more secure and less anxious. So clearly this must be happening in us too. So how can we, learn from this and decide, what kind of bigger problems can we solve? So it turns out that people have been working on this for years now, and there's this big consortium called ENCODE, called Encyclopedia of DNA Elements, and they have mapped out many different types of modifications in different kinds of cell types. And now there's many open questions based on these large data sets as to what these marks mean, how they control disease versus non-disease states, whether these marks are inherited, and so on. And this and many other questions are being solved, attempted to be solved in NCL by many different scientists. And if you like computational and mathematical biology, I encourage you to go and look up our websites. If you like computer science, then look up mine as well. And do write to me if it interests you, if you think that you'd like to do a PhD in this area at some point. And I'll stop here. I've already, I think, gone over the 40 minutes, and I apologize for that. Yeah. I'll take questions.
Yeah, thank you, Dr. Leelavati. It's really interesting. I don't want to stop it, but looking at the time constraint, we have to. So we have received some questions related to some part where one person had asked about why optimization is so important in machine learning.
So let me just go back to the slide where I showed the function. So optimization is important, but the more sort of equally important is the function that you want to optimize. Okay, so is that, the maximum likelihood function that we optimized. This is all because I can't find my mouse. So you have to bear with me with this. Yeah, this function, right? Which we plotted, right? Yeah. And I said you have to optimize this function. When you say optimize this function, I just mean you have to find the parameters where this function is the max, right? In this case, it's very easy to do it, right? You can find it right there. But in many cases, your function looks really crazy. First of all, it will not be dependent on only one variable. It will be multivariate. You can't even plot it, right? And on top of that, here you see there is one optima. Many functions that might be ideal for your problem will have several optima, right? And it may not even be continuous, right? In which case, learning the best at the top value becomes really hard. You might either end up being at a local optima, or you might there might just not be any way to find the optima at all because it's not differentiable, right? So a lot of it. So optimization and the function that your optimization go hand in hand. So oftentimes, if you realize that the function is really difficult to optimize, you might even tweak your function, which has the same characteristics. You don't really want to mess up the model, but you want to make clear that the optimization works well. So that's why optimization is very, very important. So if you don't use the right value, if you cannot find the best values, then you're just not making, it's not the best answer. So does that answer the question?
Yeah, somehow.
Okay.
I hope, yeah. Heading towards the next question, there someone had asked about what about creative words? Means if we are having some algorithm to filter mails or messages, but somebody is using some creative words to represent that spam type of thing, then what to do? Is there any way to, yeah.
This is a very good question, which is why I think, I use Gmail, so I have seen these things where it asks you, or many other email servers also ask you to say whether it's spam or not, right? And you're basically helping it to, you read it and you figure out whether it's spam or not, and you tell the server, right, that it is spam, and then it can update these creative words. Because if it sees, so essentially, if it's a spam and it's using creative words, the reason it's called spam is because it's sending it to many people, right? For you, it might look very creative, but from the server point of view, that email has gone to many people. So many of those people are going to mark it as spam. So overall, the data is multiple. So it can then update its list of words that amount to spam. But yeah, if it's the first time somebody is using some crazy, that's the problem with all machine learning, right?
So reinforcement learning is happening by adding the tags from, yeah.
Yes. So, but that needs some input also from.
Yes.
Correct. Exactly. Yeah. So what I talked about was a very simple, supervised learning essentially. But there are many more.
Yes.
Yeah.
So there was one more question that how we can start learning about attribute significance for our data. Is there any way to do attribute significance? Means something similar to feature selection or?
So one thing I would definitely advise if you're a student is to move away from tools in the beginning. Try to do this on your own, right? So first of all, the problem with these tools is they need it in a certain format, and not your method, your model, or what you have in your head may not always be translatable to what a tool might insist on wanting. Okay, so I think it's a good idea if you have a sense of what your domain wants and what kind of features should be there. You need to write your own script or own sort of method to extract those features. So that's where the domain knowledge is very important. If you absolutely have no domain knowledge, then you should look at these, so for example, the bacteria versus human DNA, right? Any biologist will tell you that's a silly, stupid thing to do, to just have ACGT, assume that it's homogeneous, right? But it's something that you can do, right? I think if you ask somebody who has no idea about DNA, that might seem like a reasonable approach to do, to have a categorical distribution of point for each, right? But as a biologist or somebody, or whatever domain you're working on, that knowledge has to come from the person doing it. Sometimes you can translate your data directly into images and feed it to deep learning and just let it do things, which is also done. So sometimes your DNA is converted to image format, and you can do all sorts of things there as well. Okay, but that's just aside. The deep learning is doing a lot of things for you.
Yes. So there is one more query that related to how to get the motif, means the way you are representing that motif image, how to get the motif from on sequence alignment or something like that.
Can you ask the person to ask it? Can they unmute and ask? Maybe.
Yeah, sure. So that is very, one can search on the net.
So if you already know what the protein binds to, like for example that I showed, where you had the the alignment already, then it's very easy. Then you do it just the way we did it for the coins, right? Where you count the number of A's C's, G's, and T's in each column and normalize. That's exactly what is done to find the motif. The trickier part is when you don't know the motif. Then you have to do some de novo motif finding, and that's a different issue. But because I don't know exactly what the question is, I should not.
Actually, that person want to know from which software you can get these motif.
Do you mean this one?
Yeah, this one.
Okay, so this one was drawn using web logo.
Okay. Yeah.
Okay, the email, the site is right there. It's very nice. It's very powerful.
Yes.
There are others too, but this is what my group has been using for a while now.
Sure. So one more question, which is from my side, related to that identical twins. Because recently we have, I had gone through one study there. They had distinguished between the way that identical twins are responding to COVID infection. Okay. There was a study which was published, I think, 4 or 5 days before. And there they had identified that there is a discrimination in the COVID response to identical twins. One get treated very easily, and another person had gone through critical care. So what could be the reason? Though you are more into computational biologist, and you have shown some data.
Presumably, if you assume that they're they're identical twins, and you assume that the DNA is also not changed, right? That means that whatever ACE2 or whatever is fine, presumably whatever it is that is fighting the infection, that machinery is different, is what we can assume, right? And the difference must not then be at the genetic level, is what I would guess. It may be at the epigenetic level. So, for example, if one of the twins was smoking throughout their life, then I might have a different outcome.
Yes.
That's just my guess. But
environmental,
pointed out, I'm not a biologist. So.
Yeah. So there are some environmental factors and other factors, not genetic factor, basically.
Sure.
They are influencing that part. Yeah.
Correct. You also hear of twins, one getting cancer and the other one not. Things like that.
Yes, like that. So that's all about the questions. And if any question is left, we can send it to you for your response later on.
The students can write to me directly if something is interesting. Please feel free to write.
So I request all participants to fill their feedback form for today's talk, and it's really important for us also. And thank you all participants as well as the team and everyone who joined this webinar. It's very interesting and very interactive webinar. I really want to thank Dr. Leelavati for sparing time from her schedule for this informative talk. And I'm very fortunate to get this insight because I'm also a computational biologist. I like the way it was presented. So thanks. So I would like to inform all participants that in the series of the webinar, we'll have more webinars in upcoming days. And for more information, just keep visiting our social media platform on Twitter, Facebook, and Insta for getting updates regarding upcoming webinars. So on behalf of all, I thank Dr. Leelavati. Now also I'd like to thank outreach team to organize this webinar and whole technical team for their support. Thank you all. Thank you for joining, and thank you, Leelavati, for this.